In deep open water, a tadpole-shaped animal beats its tail inside a transparent structure larger than itself. The structure looks abandoned and ghostly, yet it is an actively pumped feeding machine. Giant larvaceans of the genus Bathochordaeus build elaborate houses from mucus secreted by their bodies. Each house filters drifting particles that would otherwise remain too dispersed for efficient capture. The central mystery is why such a delicate animal repeatedly invests energy in disposable architecture. Tail beats pull water through layered filters, concentrating food while screening troublesome particles. As the filters clog, the larvacean abandons the house and begins another. You entered this dim current, the nearly invisible walls would surround an animal only centimeters long. The house magnifies its influence, processing water across a volume far beyond the animal's body. Its fragility is not failure, but part of a cycle linking feeding, disposal, and carbon export. The investigation follows one house from secretion through inflation, filtration, clogging, and descent. Every stage responds to particle conditions, while creating consequences for surrounding ecosystems. Direct observations reveal pumping and abandonment. Although complete construction remains difficult to watch in darkness, mainstream interpretation identifies the house as a sophisticated external filter rather than shelter alone. Its unanswered details concern formation, selectivity, maintenance costs, and changing performance across species. Larvaceans are tunicates that retain a larval-like body plan throughout their lives. They possess a trunk containing major organs and a muscular tail used to generate water flow. Unlike attached adult sea squirts, larvaceans remain free-swimming within the plankton. Bathochordaeus species occupy deep pelagic waters, where particles arrive from above and form locally. The surrounding ocean contains marine snow, microbes, tiny plankton, fecal material, and mineral fragments. Food is abundant in number, but often dilute, demanding an efficient way to gather small particles. The larvacean secretes mucus from specialized regions on its trunk. Compact material unfolds and inflates as tail-generated flow opens the house. An inner filter surrounds the animal, while a larger outer structure intercepts incoming particles. The exact geometry varies, so simplified diagrams should not be mistaken for every species. Water enters through defined pathways and moves toward the inner feeding apparatus. Tail beating provides the pump, making behavior inseparable from architectural function. When flow begins, suspended particles contact mucus meshes, whose openings exclude or retain different sizes. Concentrated edible material reaches the mouth through the inner filtration system. Oversized or obstructive particles collect elsewhere, protecting finer filters for a time. This cue-action adaptation sequence turns ordinary swimming movements into continuous water processing. The consequence is feeding access to particles too small or sparse for direct pursuit. Mucus works well because it forms light, extensive meshes without rigid mineral support. Seawater bears the structure's weight, allowing large volume at modest material cost. Transparency may reduce visual detection. Although deep predators use more than vision, the house can also separate the animal physically from some hazards and particles. Protection remains a secondary possibility, unless a specific defensive effect is observed. Filtration is the verified dominant function, supported by anatomy, flow, and captured material. Deep vehicles provide video, particle imaging, collected houses, and flow visualization around living animals. These approaches reveal function while preserving more context than rough net sampling. Vehicle lights can change animal behavior and make transparent mucus appear more solid than usual. Collection often collapses houses, destroying precisely the geometry researchers need to understand. Particle images show capture locations but cannot automatically distinguish food from rejected debris. Flow tracers demonstrate pathways, though introduced material may differ from natural particle mixtures. The combined evidence nevertheless confirms active pumping through an external mucus apparatus. One hypothesis predicts that the outer house protects inner filters by removing large clogging particles. It gains support when coarse material accumulates outside, while finer flow continues toward the animal. Another hypothesis emphasizes predation reduction through distance and transparency. Testing it requires natural predator encounters, which remain rare and difficult to interpret. A third hypothesis proposes that house size increases the water volume searched for food. That benefit should depend on pumping rate, filter resistance, and local particle concentration. If you measured only diameter, the house might appear efficient while an obstructed filter processed little water. Performance, therefore, requires simultaneous measurements of geometry, flow, and retained particles. As particles accumulate, resistance rises, and the tail must work against a worsening pathway. The larvacean may alter beating, clear portions, or eventually leave the structure. Abandonment prevents endless investment in a filter whose energetic return has declined. The animal escapes through an opening and secretes a replacement, sometimes within a relatively short interval. Reported replacement rates vary with species and conditions, so no universal schedule should be assumed. Laboratory observations can reveal sequences, but pressure, diet, and confinement may alter construction. Field records capture authentic context, yet seldom follow one individual through many complete cycles. The house's extreme delicacy also means missing details can reflect damage rather than natural absence. Researchers must distinguish a newly forming structure from a collapsing or partially eaten one. That distinction sharpens the core explanation. Disposable mucus permits rapid renewal after inevitable clogging. The abandoned house then begins a second ecological life as sinking marine snow. Loaded with captured particles, fecal material, microbes, and mucus, it can become denser than surrounding water. Descent transports organic carbon from the animal's feeding depth toward deeper layers. Some houses sink rapidly enough to reduce the time microbes have to consume their contents above. The exact sinking rate varies with load, shape, degradation, turbulence, and encounters with other organisms. As the structure descends, scavengers and microbes feed upon it, fragment it, or alter its chemistry. You might see waste, but deep communities encounter a concentrated packet of usable material. This transfer connects minute tail beats with carbon movement through hundreds of meters. Giant larvaceans can therefore influence both particle capture and the biological carbon pump. Their importance depends on abundance, house production, particle conditions, and the fraction reaching depth. One spectacular animal cannot establish the global contribution of an entire group. Long surveys and measured fluxes are needed before scaling local observations across oceans. Houses also create temporary habitat surfaces for microbes and small organisms in otherwise open water. Those passengers may consume trapped particles, modify mucus, or accompany the structure downward. The resulting packet is not inert architecture, but a changing community and chemical environment. Predators may consume the larvacean, bite through the house, or exploit concentrated particles nearby. Directly documented encounters remain limited, leaving much of this food web role unresolved. Changing surface productivity can alter the quantity and character of material reaching larvacean depths. A dense particle pulse may provide food while accelerating filter clogging. The animal responds by pumping, sorting, abandoning, and rebuilding as conditions shift. That flexibility is the principal advantage of making a house disposable. Ocean warming and oxygen change may shift suitable depths, metabolism, and particle supply. Predicting responses requires species-specific physiology rather than assuming every Bathochordaeus behaves identically. Future evidence should record complete house formation around undisturbed animals in natural pressure conditions. Three-dimensional imaging could map meshes without collapsing them during collection. Simultaneous flow and particle measurements could calculate capture efficiency as filters gradually clog. Following marked individuals could reveal rebuilding costs and behavioral changes across successive houses. Chemical analysis of sinking houses could measure how much carbon survives to different depths. Evidence for protective screening would strengthen if damaged outer filters consistently preceded inner filter failure. Evidence for size benefits would strengthen if larger houses increased net energy gain after pumping costs. Predator observations could test defense without treating absence of attack as proof. Carbon budgets must include houses that dissolve, fragment, or are eaten before reaching deep traps. The unknowns remain substantial because transparent structures vanish easily from both nets and cameras. Better observation may reveal construction rules while revising current estimates of ecological importance. At the opening scene, the larvacean still beats its tail inside walls made from secretion and water. If you follow the discarded house downward, feeding architecture becomes a vehicle for carbon export. The giant larvacean builds mucus houses because renewable filtration transforms dilute particles into food, then carries captured carbon toward depth.